Good morning. Today we'll see an important topic in muscular physiology. So we'll talk about the structure and function of neuromuscular junction. So the physiology, as well as the second part of the today's lecture, we'll see the physiological process, that is excitation, contraction, coupling. So these are two areas that we will be exploring today. So our objective today is to study the structure of neuromuscular junction and the mechanism of neuromuscular transmission, and a little bit review about the neurotransmitter, that is acetylcholine, how it is synthesized, and how it is released, and what is the effect of it, that we'll see today. Yes. So this is the diagram, a beautiful diagram of neuromuscular junction. What is neuromuscular junction? It is the junction between the neuron as well as the muscle. That is otherwise called as myoneural junction. It is the functional connection between a neuron and a muscle. So you know all this. From here you can see this is the normally the skeletal muscles supplied by alpha motor neurons. Mostly they come from anterior gray column of spinal cord, and there are large diameter ones with the myelin sheath. So from where the motor neurons take origin, and before they are all covered with the myelin sheath and plaster. So before they innervate the muscle, before that they lose their myelin sheath and they branch out profusely. And each branch in turn will innervate a single muscle fiber. So in other words, a single alpha motor neuron with all its branches and innervated muscle fiber constitute a motor unit. Now, motor unit is nothing but a single alpha motor neuron with all the branches and innervated muscle fibers together is referred as motor unit. Now, those alpha motor neurons which are having lesser diameter will give fewer branches, and the motor unit size will be smaller, and the number of muscle fibers in a motor unit may be from few to few hundred. Those motor units which are involved in fine skill movements such as drawing, etc., are the some of the extraocular muscles, etc., which are involved in fine skill movement, where the number of muscle fibers in the motor unit will be very few. But in a gross portion maintaining activity such as the muscles of the back, etc., or the girdle muscles, where there's a gross portion maintaining activities involved, where the number of fibers in the motor will be larger. So it is a junction between a motor nerve terminal with the muscle fiber. So the number of fibers involved by a motor neuron depends on the type of muscle involved. As I said, fine skill movement, few number of fibers. The gross portion maintaining activity, more number of fibers. In muscles for precision movements, few muscle fibers involved by a motor neuron. Example, muscle used for drawing, etc. In muscles used for strength or postural control, hundreds of to 1,000 fibers are involved by a single large myelinated fiber. They give more branches, and each branch will innervate a single fiber. So the motor unit will be a larger motor unit. So those are involved in gross portion maintaining activity. You can see again, these are myelinated fibers. They lose their myelination, gives branches. Individual branch will innervate individual muscle fibers. So the motor neuron with the muscle fiber. These are the muscle fiber. The nuclei they are located in skeletal muscle in the periphery, and these are the neuromuscular junctions. These are the skeletal muscle fibers. Now, in the skeletal muscle, where it is forming a connection, functional connection with the neuron, they undergo certain specialization. So now the muscle membrane, that is the plasma membrane, in this case it's called sarcolemma, now gets undergo a little bit invagination and gets thickened. The thickened portion of the sarcolemma, where the neuron is going to make a functional connection, that area is called as the motor end plate. So motor end plate is nothing but the thickened part of the muscle membrane, the sarcolemma. It is in close contact with the axon terminal of the alpha motor neuron. Now, it shows a small depression, the depressed area, and the depressed area is called the synaptic gutter, or otherwise called as synaptic trough. So, as I said, the alpha motor neuron loses its myelin sheath and divides into a number of filaments, and each filament will somewhere in the center of the muscle fiber, they make it. Suppose here they have shown so many, but it is not correct. So, only single branch will get only a single muscle fiber. More than 98% of the muscle fibers in our body, they have single innervation, barring very few, where sometimes there may be multiple innervation of a single muscle fiber may receive multiple inputs. But otherwise, in most cases, it is a single innervation. Now, as I said, the alpha motor neuron, which is a large diameter one, which loses myelin sheath and there was profuse branching, individual branches. And the terminal part of it before the impinge on the sarcolemma, this is the thickened portion of the sarcolemma, this is called motor end plate. Before it makes connection, it impinges into the trough, synaptic trough. So the end of these axons are called axon telodendria, or they are called synaptic knob or synaptic buttons. They are somewhat swollen area. In this area, suppose it is a large one, here you will find so many mitochondria indicating they are highly, that the metabolism is happening, and there are so many vesicles containing the neurotransmitter. In this case, it is acetylcholine. Now, even the somewhat invaginated or thickened motor end plate, that also undergoes further structural specialization, which are crowned into so many foldings. It's called subneural clefts. These are the subneural clefts, or some people call them the palisades. Palisades, subneural clefts are the so many invaginations which are produced from the motor end plate. These are the areas where you will find more number of acetylcholine receptors. Acetylcholine receptors are more there. These are the folded sarcolemma. So this area is the synaptic cleft. The difference between the gap between the synaptic vesicle, the axon terminal, and the motor end plate is called the synaptic cleft, which is normally 20 to 30 nanometer in diameter. So in most cases, so this entire area externally will be covered by a Schwann cell. So this area will be covered by a Schwann cell that will prevent the diffusion of neurotransmitter from this particular area. So it will sharpen the signal. So the entire area will be covered by 2 or 3 Schwann cells will come and they will cover this area. They are the glial cells they will cover. So this is the structure of neuromuscular junction. So alpha motor neuron losing myelin sheath, giving so many branches. Each branch will form connection with the center of the muscle fiber, where the muscle fiber will undergo some sexual modification. The muscle membrane, sarcolemma, undergo a little bit depression, gets thickened. This area is called as the end plate. Then the end plate further gives so many foldings that called as palisades or subneural clefts. Here, these are the, which will increase the surface area, as well as these are the sites where so many acetylcholine receptors are anchored. Now, the gap between these two is called the synaptic cleft. Now, even the terminal, axon terminal undergoes so much gets swollen, synaptic buttons or axon telodendria, where you find more number of vesicles containing acetylcholine. There may be around 3 lakh such vesicles. And they have some more number of mitochondria, so that particular area is highly metabolic. This is the structure. So each filament develops a contact with separate muscle fiber. And the terminal part of the filament shows dilatation called synaptic knob or sole feet or terminal buttons. And under the electron microscope, you can see the muscle membrane is thrown into so many folds called as palisades or junctional folds, or otherwise called subneural clefts, which increase surface area. As I said, they also enter the acetylcholine receptor. The nerve membrane in close approximation of the muscle membrane is called as the part of the membrane which is part of the nerve, which is called as the presynaptic membrane, and the membrane, the sarcolemma, which forms the depression, which undergoes specialization, that is called as the postsynaptic membrane, or otherwise called as motor end plate. The gap between these two is called as the synaptic cleft, which is 20 to 30 nanometer in diameter. Now, again, the same thing. This is the depression, this is the motor end plate. We use so many foldings. This area also, well, metabolic has so many mitochondria. Even the axon terminal also shows numerous number of mitochondria, as well as the vesicles, synaptic vesicles containing acetylcholine. The same thing enlarged here. So here you can see. This is they are the AI junctions. So these are the sarcomere, you can see. These are the nuclei. These are multinucleated structure, and they are located at the periphery. The nucleus of the nuclei of the skeletal muscles are more in number, and they are located at the periphery along the near the sarcolemma. The synaptic nerve terminal contains mitochondria and synaptic vessels, as well as synaptic vesicles with the neurotransmitter acetylcholine. As I said, there may be around 3 lakh such vesicles, and each vesicles may contain approximately 10,000 acetylcholine molecules. 10,000. And acetylcholine receptors are found near the junctional folds on the postsynaptic membrane, or the subneural clefts or palisades, and both will have presynaptic and postsynaptic membrane. Both are having ion channels. What are they? How they function? We'll see slowly. This is the same structure, which is a little bit enlarged. The axon terminal having, these are the active zones or the dense bars. These are areas where normally acetylcholine molecules will nearer to that, they will come and fuse and they release their contents. These are the synaptic vesicles, and these are the acetylcholine receptors, which are in the junctional folds, where they are more in number. They increase both surface area as well as they anchor the ACH receptors. These are the ion channels. AC receptors act as the ion channels. So the junction is covered by Schwann cell. This insulates the junctional cleft from surrounding fluids and prevents some loss of neurotransmitter by diffusion. So same thing. This is again Schwann cell. This is the alpha motor neuron synaptic cleft button. This is synaptic cleft. These are the palisades, the subneural folds with the ACH receptors. Typically, they occur at AI junction of sarcomere. Now, how the mechanism of transmission, how transmission of impulse from neuron to muscle occurs. Now, there is an intent for any muscular activity which causes the release of arrival of impulse along the alpha motor neuron. Along the alpha motor neuron, as it reaches the presynaptic terminal or axon terminal, where you will find many number of voltage gated calcium channels. So, arrival of impulse means action potential arrives at the presynaptic terminal, and which causes any action potential cause initially the depolarization, followed by repolarization. There is a depolarization. This depolarization of presynaptic membrane causes the opening up of voltage gated calcium channels. The voltage gated calcium channels open up. The calcium content within this synaptic terminal is low compared to the surrounding interstitium. So, due to the opening of the channels, the calcium ions enter inside. The calcium ion enter inside. The entered calcium does many things, of course, which are all postulates, which are not still completely proven. What it says, the calcium level will increase within the synaptic terminal. This increased calcium will activate another calcium binding protein called calmodulin. This calcium calmodulin as a complex that will phosphorylate certain kinase enzymes. The kinase enzymes. So once such kinase is snaps in, so when it is being phosphorylated, so normally the small clear vesicles containing acetylcholine, they are normally anchored to the cytoskeletal elements within the presynaptic terminal. Cytoskeletal elements like microtubules, microfilaments, etc., to which normally ACH vesicles are anchored. Now this anchoring is loosened, then they are released. So now the acetylcholine vesicles will move, they will go and bind with the specialized areas called active zones, where the acetylcholine vesicles will initially bind, then later fuse, and later they release their contents by a process called exocytosis. The whole process we normally call it as the priming and docking. How it happens, the molecular mechanism still we are studying. So we can say there is an entry of calcium which is proven and which causes exocytosis of acetylcholine containing vesicles. So the process is happening within the presynaptic terminal is called priming and docking, then release. These are three processes which taking place, wherein the anchored acetylcholine containing vesicles are released and they move to the active zones where they fuse and they release their contents, that is the acetylcholine. Now, the release of neurotransmitter from a vesicle into the synaptic club by exocytosis. Now, the release acetylcholine will bind with the specific receptors on the postsynaptic membrane. Now, acetylcholine is having different type of receptors. The type of receptor found on the junctional folds and the polysates are the muscle type nicotinic acetylcholine receptors. What is that? Muscle type nicotinic acetylcholine receptors to which they bind, which is on the postsynaptic membrane. So this receptor is a protein, it's a channel protein. Receptor is nothing but a channel protein. Now binding of acetylcholine to its specific receptors will cause certain conformational change or structural change in the channel protein. Thereby, it allows the entry of sodium from ECF into the sarcoplasm. From ECF into sarcoplasm, because sodium levels are low inside and more outside, and there is a negativity inside. Both will favor the inward movement of sodium ions through these gated channels. Why it's called gated? Because this opening and closing is gated by the binding of acetylcholine to receptor. That's why it is called as the ligand gated channel. Usually, ACH binds to the ACH receptor. This is a channel protein which leads to opening. So this is called as a ligand gated sodium channels. There is an entry of sodium via ligand gated sodium channels. Now there is influx of sodium which causes movement of positive charge from outside to inside. So temporarily inside the membrane, now it becomes positive. Earlier it was negative due to movement of positive charge sodium ions inside. It becomes positive inside. That means the membrane gets depolarized. This change in the membrane potential, which is due to influx of sodium via ligand-gated sodium channels, is called end plate potential. This end plate potential is an example of graded potential, which is a non-propagated one. So normally, the number of ACH vesicles which are released will be around 125 for each signal. Every time the acceptor reaches the presynaptic terminal, they release about 125 such vesicles, each having 10,000 such ACH molecules. So invariably, the number of ACH release will be more than sufficient to produce the end plate potential. This end plate potential will invariably reaches the threshold. Why it is controlled? Because of release of more number of ACH containing vesicles. In turn, each will have about 10,000 molecules. So now all end plate potential, which are the local depolation which are happening in the motor end plate, they will summate. Now the summated potential usually reaches threshold. Now an action potential will be fired. That means that particular area becomes less negative or become positive. Now due to this, there is a next step is the opening up of the voltage gated sodium channels. Initially, there is entry of sodium via ligand gated channels, which leads to EPP, that's the end plate potential. This EPP, when it reaches threshold, now it causes a local depolarization, and which causes the, or which makes the membrane potential move towards from resting level to a firing level or to a threshold level. Once it reaches threshold level, now an action potential will be fired. This action potential is fired due to sudden influx of sodium inside via, this time via the voltage gated channels, Now that's the development of action potential at the motor end plate. Now this action potential spread on either side of the particular neuromuscular junction. Along this sarcolemma, it will be propagated. Now, you know, the sarcolemma will make so many invagination, and they make inroads into the sarcoplasm by way of T tubules. So the action potential reaches the T tubules also, where it is going to causes the EC coupling, excitation contraction coupling, that we'll see 3 minutes later. Now you know the whole sequence. That is arrival of impulse along the alpha motor neuron. Now, due to this voltage or due to this wave of depolarization, there is an opening up of voltage gated calcium channels at the presynaptic terminal. Now, movement of calcium inside, which leads to priming, dogging, and exocytosis of vesicles. Now, release of acetylcholine. Now, it binds to the specific muscle type reporting ACS receptors. Now, this binding causes conformational change, thereby leading to opening up of the sodium channels. In this case, it is ligand gated sodium channels, which causes the positivity inside and the development of end plate potential. This end plate potential, once reaches the threshold, it fires an action potential by sudden influx of sodium via the voltage gated channels. This is the events. Now, once the action potential is generated, which is a propagated one, it spreads on either side of the sarcolemma, on either side, and which also passes into the muscle by means of T tubules. So this is what happens. The action potential arrives, calcium influx, release of acetylcholine, and binding to the receptor. And the release and binding to the specific receptors. Now, which leads to the development of initially EPP, then later action potential. Now, once action potential develops, it also spread along the sarcolemma. It also goes into the T tubules, where it causes further events that we see under what we call EC coupling, with excitation contraction coupling. This is what happens. Now you can see the animated thing, how it goes. Now spread of action along the sarcolemma. Now there is a contraction. You can see once again, you can follow the course. There is arrival of impulse and release of acetylcholine and EPP, and action potential, spread of action on either side, and again release of calcium and the active myosin interaction. This is how the sequence keeps going. So the same thing, the axon terminal, arrival of impulse, and again calcium influx, and there is a release of acetylcholine from these vesicles, binding to the receptor, and again development of EPP and development of this EPP, then development of action. Once it is the threshold reaches the action potential and expressed along the sarcolemma into the T tubules. These are the events which happen at the neuromuscular junction. Now, what happens to the release of acetylcholine? Acetylcholine synthesizes in cytoplasm from acetate and choline, and which is stored in the synaptic vesicles, and with the help of an enzyme called choline acetylase. And whenever the nerve impulse passes the presynaptic vesicle to the synaptic membrane, it causes the perimeter by increased permeability of calcium, and And now entry of calcium causes release by exocytosis, and most of the ACH released will be destroyed in that area by presence of an enzyme called acetylcholine esterase. Acetylcholine esterase is an enzyme which will allow acetylcholine to act only few milliseconds, not more than that. Sooner they will be destroyed by an enzyme called acetylcholine esterase, and it will be cleaved into choline and acetate. Now, some of the acetylcholine will diffuse back into the nerve terminal, and they will be taken up by the nerve terminal for further synthesis. So it will be broken down into choline and acetate, and choline will be taken up by the nerve terminal, and acetate will enter the bloodstream again, reach the nerve where it is used for further synthesis. So the effect of acetylcholine at the nerve terminal is very, very short-lived. It doesn't act forever. Only very few milliseconds they remain, and after that they will be cleaved by the enzyme, powerful enzyme, acetylcholine esterase. This is the effect of ACH, how it is synthesized, and where it acts, how it acts, and what happens to it later. Now there are certain drugs that will act at the neuromuscular junction. Now the drugs can be of various types. Number one, there are certain drugs. They will inhibit the release of acetylcholine from presynaptic terminal, like conotoxin and alpha beta etc. Sorry, botulism toxin, etc. They prevent the release of acetylcholine from the presynaptic terminal, and some of them they will bind with the acetylcholine receptor competitively, thereby they will interfere with the activity of the acetylcholine. And certain other blocking agents, they will inhibit the acetylcholine esterase, thereby temporarily they will cause persistent depolarization leading to muscle spasm and later. And some of the drugs are poisons, they may bind with the acetylcholine esterase irreversibly, thereby leading to neuromuscular blockade, and because that also leads to persistent depolarization, and it will cause stoppage of transmission across the muscle, so muscle spasm. So there are various agents are there. Some of them they will act at the presynaptic terminal level itself. Even some of them they will inhibit the sodium channels like tetrodotoxin. They will block the sodium channels. Thereby they can also interfere with the transmission across the NMJ. So the drugs that they block transmission called neuromuscular blockers, and there are certain drugs they will stimulate by anti-choline esterase, because they will bind with the choline esterase, so they are anti-choline esterase, so that thereby they inhibit irreversibly or as well irreversibly. Thereby, acetylcholine will remain in the particular area for a longer period, so that leads to persistent depolarization for the blockade. Neuromuscular blockers like botulism toxin coming from bacteria Clostridium botulinum, which inhibits the synthesis and release of ACH from presynaptic terminal. This leads to paralysis, and it is sometimes used in the treatment of stiff neck as well as laryngospasm, wherein botulism toxin is given as a drug for relieving the spasm, because it prevents the spasmodic contraction of the muscles in the particular local area by inhibiting the release of ACH. Like say the alpha bungarotoxin, it is derived from the venom of kite and binds with receptors irreversibly, and it causes the paralysis by substrate complex. It doesn't allow, it binds with the ACH receptors irreversibly, so that ACH cannot bind them, it cannot mediate the neuromuscular transmission, thereby it causes paralysis. These are the picture of kite. The venom contains alpha bungarotoxin, which causes neuromuscular blockade. Now there are certain other drugs, even the curare is the drug normally or in the arrowhead poison used by red Indians for hunting, etc. They use on the arrowhead the plant derived product. Now we have the synthetic tubopurine and galamine. These are they combine with the receptors reversibly. Because for short-lived, they're actually short-lived. Sometimes they will inhibit the choline esterase by competitive inhibition and used in surgery and electroconvulsive therapy. In psychiatry, they give what ECT, electroconvulsive therapy they give. So in such cases, in order to have the in surgery to have a free feel, so to prevent the muscle spasm, they give this thing as a centrally active muscle relaxants because they compete with the acetylcholine receptors competitively. Of course, this binding is irreversible. After some time, acetylcholine will have its action after some time. Now, the other drug like acetylcholine, nicotinium also blocks transmission by persistent depolarization. They cause persistent depolarization. Thereby, once the muscle is persistently depolarized, there will be a blockade of the muscle junction. So that's another group of drugs. They cause persistent depolarization. They mimic the action of acetylcholine. Thereby, they will cause persistent depolarization. Because they cannot be cleaved by the acetylcholine esterase. Now there are some drugs that will stimulate transmission. They are called as anticholine esterase. Because why they stimulate? Because they doesn't allow the acetylcholine the esterase to be there in the vicinity, so that the action is the action of acetylcholine is for a prolonged period. Like neostigmine and physostigmine, the action is temporary, reversible, and prevents destruction of acetylcholine by choline esterase. Because choline, they are anticholine esterases, so that it inhibit the activity of choline esterase. Thereby, acetylcholine will remain for longer period there, so that they can cause persistent depolarization. Now, this is diisopropyl pyrophosphate and as well as tetraethyl pyrophosphate. They come from the organophosphorus, like poisons, organophosphorus compounds, like most of the insecticides, like parathion, malathion, etc. These are the active compounds in some of the nerve bases also. So, these kind of compounds, they inactivate the acetylcholine esterase irreversibly, so that they becomes fatal. So they completely, but whereas the previous one, neostigmine, physostigmine, they are reversible, temporary blockade, but they will cause permanent blockade, thereby it causes death. So that's why some people, when they attempt with organophosphorus compounds, most time it is lethal because it causes the it inactivates acetylcholine esterase permanently, thereby causes initial the spasm, later there will be neuromuscular blockade and it will be fatal because they cause spasm, even spasm of laryngeal muscle, etc. can lead to death. So now we have seen what is neuromuscular structurally and how the transmission occurs, and as well as what is the rate of acetylcholine, as well as what are the various neuromuscular blocking agents and what is the mechanism of action that we have seen. So that completes the discussion on neuromuscular junction. So now we'll see another subtopic that is the coupling. Before we go into that, there's one applied aspect, that is myasthenia gravis. Myo means muscle, asthenia means weakness. It is an autoimmune disorder, and there's the production of autoantibodies against one's own ACE receptors. So ACE receptors will be attacked by the autoantibodies. So available ACE receptors will be very minimal. So progressively the muscle becomes weak. In the morning they will be all right. As the day progresses, they become more and more weak. They generalized weakness, and once it is untreated or it is not treated properly, it can lead to fatality. Because most commonly affected muscles are the extraocular muscles, facial muscles. To start with there, and interfere with swallowing, mastication, etc. And severe cases, the patient may remain bedridden or die due to respiratory muscle paralysis. So they can be easily treated with the drug called neostigmine. It's an acetylcholine esterase inhibitor. So thereby acetylcholine will not be acting at the neuromuscular junction. So the available ACH will be acting on the available ACH receptors, so that it will increase the choline, the acetylcholine levels there. Thereby, it will activate the available ACH receptors, because there will be few in number because of attack by the auto antibodies. So only very few ACH receptors will be there. They will be activated by the available or high concentration of ACH available in the neuromuscular junction. So slowly the muscle weakness can be relieved. Muscle spasm will be relieved due to the action of neostigmine, like a magic molecule. Once it is given, it will give relief. So that itself indicates the diagnostic and treatment, like the patient is suffering from myasthenia gravis. So it is usually asked start course, try to read well this one. So this is summary. There is anatomy. We have seen secretion, acetylcholine. We have seen and receptors. We have seen impact potential, how acetylcholine generated in upper aspects. We have seen about the myasthenia gravis and NFJ blockade. So now the next we'll see the EC coupling or excitation contraction coupling. So here we want to see the mechanism of EC coupling. What is the mechanism? Now, once again, we go back from the neuromuscular junction. The mechanism of nerve, alveolar impulse, vesicles move and attach to the membrane and release, and AC is combined with the receptor, causes EPP. Then later, the action potential will be propagated along the sarcolemma, and it also moves into the T tubules. These are the C tubules. And now schematically, level of axial potential and release of ACH in the presynaptic terminal, and axial potential propagates through T tubules. Thus, this acetylcholine binding to the specific receptor, muscle type nicotinic receptors, which is channel protein, causes the inward movement of sodium, which causes initially EPP, later axial potential, and this axial potential spreads along the T tubules, where when the axial potential moves along the T tubules into the sarcoplasm, where you will find another type of calcium channel, which is voltage gated calcium channels, which is otherwise called as dihydropyridine. Dihydropyridine type of calcium channels, which are voltage gated. So they will open only when there is a depolarization wave, otherwise they don't get open. So when the action potential moves downward into the muscle along the T tubules, due to this deep wave of depolarization, now the dihydropyridine type of calcium channels open up. Now this opening up dihydropyridine calcium channels, in turn it will be hooked to the another type of calcium channel, which is there in the cisterns of L tubules. This is the T tubule. These are the cisterns, somewhat swollen parts of the. These are the L tubules, longitudinal tubule. This is the transverse tubule. These are the part of the endoplasmic reticulum or sarcoplasmic reticular network. So this is the T tubule, where you will find the dihydropyridine type of calcium channel, which are voltage gated. Once it's open up, it will automatically open up the ryanodine type of calcium channels, which are there on the cisterns of L tubule, which is non-voltage gated. The opening up of ryanodine type of receptor is purely non-voltage gated, and due to the opening of dihydropyridine. Once dihydropyridine calcium channels open up, automatically it will open up the ryanodine type of calcium channels. It will cause the outpouring of calcium from the cisterns into the sarcoplasm. So calcium released from sarcoplasm reticulum into the sarcoplasm. So that's what happens. Now, there is an electrical event. That is the arrival of impulse, release of neurotransmitter. Then again, once again, development of EPP, action potential, spread. Then again, movement of calcium via initially T tubule dihydropyridine receptors, as well as outpouring of calcium from the sarcoplasm tubular into the sarcoplasm via ryanodine type of calcium channels. These are the events which will take place in the muscle. Now, calcium is released. The calcium content within the sarcoplasm or within the muscle increases, 100-fold. Initially, at rest, the calcium levels are very low, which doesn't cause us the interaction of calcium with the muscle protein troponin. The interaction with calcium will occur only when the intracellular calcium levels increases. So this is what happens now due to arrival of impulse from the nerve terminal. Now there is a neuromuscular transmission, spread of transmission along the T tubule, release of calcium, increased intracellular calcium. At this increased intracellular calcium level, now calcium ion will bind to the troponin. Now troponin is having, it's a button-like molecule which is get attached to the actin on one hand and tropomyosin on the other hand, and it is also having binding site for calcium. The troponin is having three subunits. There is troponin I, with which it will attach with the actin. There is the two strand of F-actin molecules, fibrous actin, and with the T subunit, which will get attached to the tropomyosin. It's again filamentous protein, normally covering the active sites of actin. Now it is also one more subunit, troponin C, with which it is having affinity for calcium. This binding of calcium to troponin C occurs once calcium levels becomes more, not otherwise. So now that's what happens. Calcium levels will become more. So you can see what happens in arrival of impulse. Now this is the dihydroprotein channel. This is the ryanodine channel. That's outpouring of calcium. Outpouring of calcium. Arrival of impulse. Opening of dihydroprotein channel. Now this is the ryanodine channel. So there's outpouring of calcium. So this is the sequence which happens. You can see from each one. Arrival of impulse. Now opening, outpouring. So this cycle continues. So every time whenever impulse comes, there is a release of outpouring of calcium from the sisters of L tubule. Sisters of L tubule. This is a T tubule. This entire section is called the triad. So a central based T tubule and L tubule sisters on either side. This section is called the triad. So they causes the release of calcium upon the impulse arrival. Now that's what happens here. There's arrival impulse along T tubule. Now entry of so the calcium the dihydroprotein channel. This is the ryanodine type of channel from where the calcium is outpoured. Calcium levels increases. It will bind with the troponin C. Troponin C. Now once calcium binds with the troponin C subunit, now which causes a conformation change in the troponin molecule. Thereby it swirls, that means it rotates. Thereby it will drag the tropomyosin along with it. It drags the tropomyosin. It's a filamentous protein. Normally it is placed along the actin filament. Now the active sites on actin are exposed due to dragging of tropomyosin. Now active sites on actin are exposed. Now it is ready for interaction with the myosin heads, which is having active site for having the binding sites for active sites on actin. So that leads to the further muscle contraction. You can see due to, again, this is the neuromuscular junction, EP motor end potential or EPP, end potential, axial potential. This is the dihydroprotein channel, which again opens the ryanodine channel, outpouring of calcium. The calcium will bind to troponin C. Now this is the tropomyosin. Now tropomyosin will be dragged along with it. Now active site, now in this case it is covered. Now it will become uncovered. Now myosin head will attach with the active sites on actin. Now see, you can see it will bind. Now it will, the hinges in the myosin head will undergo what we call power stroke. Thereby active filaments will be moved towards the center by power. Now this is called cross bridge formation. This cross bridge undergo cycling. Axial potential T tubule alters conformity dihydroprotein receptors, and this opens the calcium channels, and calcium binds to the troponin, allowing strong actin myosin binding, the myosin heads execute power stroke. Now actin filament slides over the center of the sarcomere, that is the muscle contraction. Now there is an electrical event, that is the action of the arrival of action potential and its transmission spreads along the G tubule. That's what the electrical events. Well, what is happening finally? There is a muscle contraction. That's the mechanical event. So on one side, the electrical event is converted into a mechanical event. That's why it is called as a excitation, contraction, coupling. Electrical events culminating or ending in mechanical activity. So now in this case, the calcium ions act as the coupler molecule. So electrical events finally leading to a mechanical activity. So now you can see the distance between Z lines will decrease at the sarcomere. Distance decrease due to continuous cross-bridge recycling. Each time active myosin head will attach to active sites and tilt it toward the center, again detaches, and again once again attach with the next active sites. Thereby every time the active filaments will be made to slide over the myosin filaments towards the center. That's what the muscle contraction process is. Now, electrical events, that is the arrival of impulse along the alpha motor neuron, leading to muscle contraction, that is the mechanical event, and both are coupled together by a calcium molecule, that is the coupler molecule. So this event, the excitation leading to contraction, this is called as the EC coupling or excitation contraction coupling. Now you can see this is the myosin head. It binds to active sites and actin. Normally, the active sites will be covered by tropomyosin. The tropomyosin uncovers this area because of calcium binding to troponin C. Now you can see every time, initially it binds with one and tilts towards center. Next time it gets released and binds with the next active sites. Like this, the cycle continues. See, like next time here. Every time the cross-bridge recycling undergoes, every time it replaces the oxygen ATP-based activity in ATP-based. And whenever ATP binds, the affinity gets decreased. Once the ATP becomes ATP and PI, because of the myosin head itself having ATP activity, so it have free energy, and again, once again, the power stroke will continue. The myosin head swings over and binds weakly to a new active molecule, and the cross bridge is now at 90 degree related to the filaments. So every time it binds and again tilts, binds, tilts, like this it goes. So which produces, every time it produces a power stroke, and there is a sliding of active filaments over the myosin filaments, and leading to the decrease in sarcomere length. We call it as the muscle contraction. So you can see this is the myosin head. Initially, it is here. The ranges are like this. Now attaches, then again tilts. Now it is 90 degree. Again slides. Next time when it becomes like this, it will attach to the next active site. Like this cycle continues, so that active filaments are made to slide over the myosin filament. You can see here what happens. So this is the tension developed. Whenever the active filaments are made to interact with the myosin filaments, then the number of filaments are more, tension developed will be more. Whenever sarcomere length increase, the tension will be less. It is based on number of degree of overlap. So the distance between two Z lines decreases in muscle, and that's what we call it muscle contraction. So how muscle gets relaxed? No more impulses. What will happen now due to the presence of calcium ATPase? Calcium ATPase, once again, the calcium ions are pumped back into sarcomerous reticulum. Once sarcoplasmic calcium levels become low, now the calcium will be released from the troponin C. Now once again, the troponin undergo sexual modification. Thereby, the tropomyosin will once again cover the active sites on actin. So binding of myosin heads to actin will be prevented. So there is no contraction. The muscle undergo relaxation. The reverse set of events will take place during relaxation. In relaxation, all the calcium ions will be pumped back into the sarcoplasm. Sarcoplasmic reticulum. Initially, it is released. Now they will be pumped back into the reticulum. Thereby, the intracellular calcium level will be low. It will prevent the interaction between actin and myosin filaments. So the muscle will undergo relaxation. The active sites and actin will be covered by propomyosin. So the muscle will undergo relaxation. So calcium ions are pumped back into the cisternae. Calcium ions concentration around the filaments will decrease. The propomyosin moves back to their original position. Myosin accept ATP molecule. Now once it accepts ATP, it will detaches itself from active sites and actin. The cross detaches. Propomyosin covers the active sites. Interaction between these two will ceases. The muscle relaxes. These are the events which take place in the muscle relaxation. Now you can see this is the walk along theory or sliding filament theory. So here, imagine the myosin head doesn't move anytime. It is the actin filament which is moving inward. So now, actually, when you see, as if the heads are moving along the active sites and actin, that's why it's called as a walk along theory of muscle contraction. It's all the perspective what we see. You can call it the walk along theory, or you can say the sliding filament theory, or otherwise some people call it the ratchet theory. All molecular mechanisms are one and the same. The active sites and actin are exposed by dragging up propomyosin by calcium interacts with the proponin C. Now the myosin will interact, it will tilt the active filament towards center, and after accepting ATP, once again detaches and attaches to a new active site and actin. Every time it will move the active filaments towards the center, like this one we are seeing. So the active filament will move towards the center. The sarcomere length will decrease. We refer as the muscle contraction. So now the entire event we have seen about what is the muscle junction, what are the various structure, how transmission occurs that we have seen. The next case, how this transmission leading to muscle contraction that we have seen as a EC coupling. So now this is the motor unit, a single alpha motor neuron with all the branches removed in muscle fiber constitute a motor unit. Now rigor mortis, this is the rigidity of muscle after death, which is due to lack of ATP. Now you know ATP is required for two things. Number one, for detachment of myosin head from active sites and actin requires ATP. Next also, for pumping back of calcium from sarcoplasm into the systems also requires calcium. And after death, there is no metabolism, there is no ATP. When this thing doesn't happen, the actin and myosin will bind with each other irreversibly, and they remain in abnormal, fixed and resistant way. So that's why it leads to spasm or muscle contraction after death. That's called rigor mortis. And it will happen, you can say 1 hour after death, and is complete within 3 hours. And it starts from lower jaw, base, neck, and finally the lower extremity. It disappears after 24 to 30 hours due to autolysis of muscle protein. And it is of medical importance to know the time of death. And thereby it can help in the diagnostic criminals. And there are some factors which will affect the rigor mortis, that is muscular exercise. Suppose there's a violent fight before death, then again the rigor mortis will occur very early because ATP will be consumed during the fight reaction itself. So, and sometimes the high temperature, chloroform, pepper, etc. So will hasten this particular process. So with this, we'll complete day and prepare well for the weekly assessment.